Okay, good afternoon, everyone, and welcome back. So this afternoon, we're going to start our discussions about bacterial disease and bacterial causes of disease. I just want to remind you, we have mentioned it before, that there are various ways in which bacteria can actually cause disease. The vast majority of bacteria, of course, in the world do not cause disease at all. It is a minority of bacteria that actually do cause disease. Those that do so by a variety of mechanisms. So there are various mechanisms of pathogenicity. The first is that they cause direct damage to the organism. They cause direct damage to the cells. Even their adherence to cells may be damaging, but they can also impair the functioning of organs, etc., just by their presence. But bacteria, the second way in which they often cause disease is that they release toxins. They release toxins, they release substances which are toxic to the organism that is hosting them. And this is something which we frequently see. We'll hear about, referred to several times. Those toxins are often referred to as exotoxins when they are actually dissolved into the body fluids, for example, released from the bacterial cell that dissolve in the body fluids, and may have their activity far away from the site where the bacteria are colonizing. Other toxins are referred to as endotoxins, and those are often components of the bacteria themselves, which when they go into circulation cause a reaction to their presence. And this highlights the fact that very often what happens in bacterial infection is that it excites an extremely powerful immune response from the host. And that immune response itself is in fact damaging, is virulent, and causes damage to the host. So there are various ways in which bacteria may cause the symptoms of disease in the host. In general, remember that we are very much protected by colonies of our own microbiota, which are present at those areas of the body which we might regard as portals to the interior of the body. So, for example, the mouth, the nose, the anus, the rectum, the urinary tract, the reproductive tract, the vagina. All of these are portals to the interior of the body. And something that characterizes all of them is that they have their own microbiota, often a very rich microbiota, which is completely harmless to the host and which is in fact protective. We've talked about this before. So today we're going to start talking about how, in fact, bacteria can sometimes breach these portals. And the first set that we're going to talk about are the portals of the respiratory tract. And these, in fact, most acquire pathogenic bacteria usually by airborne transmission, often in droplets, but sometimes also through fomites or through direct contact with somebody who is actually infected. So, characteristic of these sort of portals. Look, for instance, at our mouth, nose, and all of the structures of the upper respiratory tract. Mouth, the nose, the sinuses. These are all invested with a mucous membrane, much of it ciliated to move mucus. In the upper respiratory tract, mucus is in general moved to the nose, where we blow it out. And that's movement of the mucus. The mucus traps particles, it traps bacteria, but it also supports the microbiota. And the upper respiratory tract has a very rich microbiota. And that, if you think just about the mouth, the nose, the sinuses, these all have an actual coating of bacteria. The mucus serves to limit the population size, but the microbiota may be extremely rich. There are many, many species there, and they coexist completely harmlessly, in fact, probably beneficially with the host, and they crowd out pathogens. And they may, in fact, some of them produce antibiotics to limit the growth of pathogens. So the possession of that healthy microbiota is in itself very protective. The lower respiratory tract is a bit different. This is not to say that the lower respiratory tract is sterile. It is not. You can, in fact, find bacteria even deep within the lower respiratory tract, but they are very few in number, and it is not a diverse population. And the lower respiratory tract has a very active mucociliary escalator. Right from the very smallest alveoli in the lower respiratory tract all the way up to the upper throat, there is a continual stream of mucus, a sheet of mucus being moved up, which is continually swallowed at the top of the throat. And that mucus traps particles brought down through the bronchi and on inhalation, for example. And it keeps the lower respiratory tract largely pretty sterile compared to the upper respiratory tract. It is not entirely sterile. But bacteria simply, under normal circumstances, simply don't have an opportunity to grow to such an extent that they could cause disease in the lower respiratory tract. So just remember the various structures. Remember the differences between the upper respiratory tract and lower respiratory tract, and between the nature of their clearance and also the nature of the bacterial populations that are normally there. So this here are just some pictures of this mucociliary clearance mechanism, especially from the lower respiratory tract, but it also applies to the upper respiratory tract. Now, the other thing that I need to mention, which we haven't talked about before, is that throughout the respiratory tract, throughout the mucous membranes of the respiratory tract, there is extensive immune tissue. It is tissue dedicated to the immune system. And these tissues often are referred to as MALT, M-A-L-T, mucus-associated lymphoid tissue, mucus-associated lymphoid tissue. And these occur throughout. There are some actual immune organs which are present in the upper respiratory tract, especially the tonsils and the adenoids are, for example, are dedicated immune organs which are there to deal with, especially with pathogen brought in through inhalation and through swallowing. But throughout the respiratory tract, in the mucous tissues of the respiratory tract, there are patches of this MALT, M-A-L-T, throughout, which are all dedicated to surveillance of what is passing through and to provoking an immune response if something arises which looks like antigen to the immune system. So there are a whole lot in those. You can think of these patches of MALT tissue being like flattened lymph nodes in a way. And they are extremely efficient at surveilling what is happening in the respiratory tract and responding to it. Nonetheless, these systems do fail. We know that. We know that, for example, they're not good. They're not particularly good at dealing with a high dose of viral pathogen. This is for the start. We've already talked about viruses. What happens often with a viral infection, for example, is that these mechanisms become overwhelmed and they become damaged and impaired. And even if the person recovers from the viral infection, often it predisposes, for example, to a bacterial infection. Here is a typical one. This is Staphylococcus. And Staphylococcus is, many of them are normal members of the skin microflora. But under certain circumstances, especially in the respiratory tract, they can, in fact, act as opportunistic pathogens. So especially when there is some impairment of these protective mechanisms, bacterial infection may also result from a very high dose of bacteria being taken in, and the systems are just simply not able to deal with it. There are several diseases whose whole pathogenesis depends upon being taken in through the respiratory tract. So, and we're going to talk about several of these. Okay, so as we go through all of these diseases, what I advise you to do is make a note of the name of the causative organisms, look up those organisms in your textbook, make a note of their gram status, their aerobic status, whether they're obligates, anaerobes, or facultative anaerobes, etc., and make a note of their principal pathogenic mechanisms, and then the symptoms, etc., that they cause. So keep a record in a sort of a tabular form, if you like, of these different organisms that I'm going to talk about. So we'll start off with some that are really quite familiar to you, and I'm quite sure that some of these are things which you have had. And the very first, of course, is a bacterial sore throat, probably one of the commonest diseases of the respiratory tract that any of us have. So it's really an infection of the upper respiratory tract that we're going to talk about first of all. The most common cause of the sore throat is an organism called Streptococcus Pyogenes. So just here, abbreviated S Pyogenes, is Streptococcus Pyogenes. Streptococcus Pyogenes is actually also a member of our microflora on our skin, but it is the commonest cause of the disease strep throat. And as strep throat, when you suffer from strep throat, you often acquire it as a result of viral infection. It may appear that it's coming simultaneously with a bad cold or with flu, but it actually comes in sequence. It is often that the tissues of the upper respiratory tract are damaged and predisposed to the strep throat. It can develop incredibly quickly. So here, have a look here, and you can see the red, inflamed back of the throat there, the pharynx there, and tonsils. In this case, the person has their tonsils. And these white parts here, this is actually dense mixture of bacteria colonizing the surface of the tissues, and also probably some immune response and pus and stuff may be present as well. Very, very painful sore throat, and often accompanied by a fever as well, which is not always a hallmark of disease. But in this case, very frequently the strep produces a fever as well. It's very easily acquired through respiratory droplets. When people around you have strep throat and are coughing, you may acquire a very high dose of a bacteria simply through breathing in other people's droplets containing the bacterium. So these streps, the Streptococcus, especially Streptococcus Pyogenes, there are many different strains, and they have different virulence. And the first thing about them is there are various mechanisms that the different strains have. And the ones that we are most interested in are a group called the GAS, G-A-S the Streptococci. I'm not going to go into all of the why they call that. GAS stands for Group A Streptococci. And these Streptococci are beta hemolytic Streptococci. Remember, hemolysis is that phenomenon that we observe where, for example, when we plate Streptococcus onto blood agar, you look at its growth, you see that around the colonies it produces a complete clearance of the blood corpuscles in the agar. That you need to realize is a reflection of the fact that these bacteria are releasing exotoxins, which not only break down hemoglobin and are able to use, but they will break down red blood corpuscles. The implication of that is very serious because it suggests that this is one of the reasons there is such a strong tissue reaction to these bacteria. They're actually damaging the tissues around which they are doing on the mucosa of the throat exactly what they are doing in the blood agar plate that we observe, right? And they're breaking down cells and utilizing the interior of the cells as nutrient. So there are certain strains which can go on from this ordinary strep throat to produce other disease symptoms. And the first of these is a group of these GAS, Group A Streptococci, which produces this substance called Streptococcal Pyogenic Exotoxin. And Streptococcal Pyogenic Exotoxin, or SPE, is the cause of a generalized systemic illness, which starts with that sore throat, but then spreads through the whole of the rest of the body. And it is the cause of the symptoms of generalized inflammation and production of spotting all over the body, which gives this disease its common name, which is scarlet fever. Now, it's not a matter of the bacteria from the throat spreading through the body. It's that they are releasing this as an exotoxin, which goes into circulation and causes these symptoms. We also say that this toxin is erythrogenic. That is, it causes these spots. It causes the blood vessels in the surface of the skin to react so that they produce this spotting, which is characteristic of scarlet fever. And it also characteristically it produces a swollen, a very red, inflamed tongue, which is called, the symptom is called strawberry tongue, and that is characteristic of scarlet fever. Scarlet fever used to be a very common childhood illness, and it was in fact extremely dangerous. Many children died of scarlet fever for reasons that I'll discuss in a minute, because it can progress from here to another set of symptoms as well. Scarlet fever on its own is a fairly dangerous disease, now rather uncommon, because strep throat is so easily treated with antibiotics, responds very quickly to antibiotics. So those are the symptoms of scarlet fever, with this typical rash. But there is a second mechanism which strep can have. This is not to say that all strep has this, produces this disease mechanism, but some do, and some of them that produce the scarlet fever can also progress to this set of symptoms. And this is this now becomes an extremely dangerous situation. Again, in the old days, this was fairly common, especially in children, and was a higher cause of mortality in infants and young children. This is the disease of rheumatic fever. And rheumatic fever often began with a sore throat, first manifested as scarlet fever with generalized rash, etc., but then progressed. And it produces pain in the joints, and then, unfortunately, very often would progress to diseases of the heart. And these symptoms are produced by a very strong reaction to the proteins which the bacteria use to adhere to the cells that they infect. These are called the M proteins. And the M proteins are the proteins that are sticking the bacteria to the cells, especially of the upper respiratory tract. Unfortunately, the M protein looks very similar to many of human proteins. And the immune system, in reacting to the bacterial M proteins, sets up an autoimmune response to the body's own M proteins that look like M protein. And these are especially manifested by inflammatory conditions in the joints and then in the tissues of the heart as well. And this could produce permanent sequelae of heart damage, especially of damage to the valves of the heart. The valves of the heart become extremely inflamed, thickened, and the tissues become seriously damaged, and the heart valve doesn't function properly anymore. So that was the one set of sequelae to this acute rheumatic fever. The other is a very, very dangerous condition called glomerulonephritis, which is inflammation of the kidneys, again through an autoimmune mechanism, which produce serious kidney damage and even kidney failure. So this group of streps, they can produce this range of symptoms. They don't necessarily produce only a progression. Sometimes they do. Other times you may have a direct progression very quickly to rheumatic fever. Very, very dangerous, but of course now, as soon as a strep throat is diagnosed, then the person will be treated, the kid will be treated with antibiotics, and this can be avoided. So here is another of what used to be the extremely dangerous respiratory tract bacterial infections, especially in children. And this is the disease of diphtheria. Diphtheria now is seldom seen. Fortunately, there is a very good vaccine, which I'm sure you've all had. And diphtheria now is really, really uncommon. Diphtheria used to be extremely common, and it was one of the reasons that most Victorian families could count on losing at least one or two children. That is how common and how serious it was in the United States. It was a very, very common cause of infant and child mortality. And it's caused by this a bacterium called Corynebacterium diphtheriae. For all of these bacteria that I'm telling you about, please learn the name. Please write it out and learn to spell it and to say it. Corynebacterium diphtheriae. It's one of the most important disease-causing bacteria in human beings, and in developing countries where vaccination may not be common or easily available, it is still a major cause of infant and child mortality. So the Corynebacterium, they have strange, it's a rod, and it has these strange characteristics that when it divides, the bacteria arrange themselves into rows where they sit like this, and it's called palisade layer. Again, it's acquired by respiratory droplets transmission, and rather easily acquired too. It could be epidemic in situations like in schools and places like that. There's a very interesting situation here, because the Corynebacterium just on its own It can produce an infection, but the infection tends to be self-limiting unless the Corynebacterium has previously been infected with its own virus. This particular virus is a lysogenic virus. Go back to the virus life histories that we talked about for the bacteriophages and look at see what was a lysogenic. A lysogenic virus was one which integrated with the host DNA, put its genes in there, and just sat silent and didn't damage the functioning of the host bacterium. Now, in this case, the Corynebacterium diphtheria acquires a lysogenic virus, which has, as part of its genome, a set of virulence factors. And these virulence factors actually affect the mechanisms of translation of the host cells. So they impact the functioning of the host cells. And this results in the production of a very strange structure on the host cells. The Corynebacterium bacteria stick to the cells. They impair the functioning of the mucous membranes that they're adhering to, and they cause the production of a thick mucus, of a very, very thick mucus, which mixes with the bacterial cells and with dead mucoid tissue to form a thick layer called a pseudomembrane. And this protects the Corynebacterium, which are facultative anaerobes, and it isolates the Corynebacterium from immune response of the host. But at the same time, it causes intense inflammation below in the mucous membrane. So what happens now, I have a look, really careful look here. What you look at, here's the uvula of this child. Here's up here, this is the epiglottis here. And right here, you can see, first of all, that there's inflammation. So the epiglottis is already swollen. Here's where the Corynebacterium is causing the formation of a pseudomembrane. That pseudomembrane begins to intrude across the opening to the trachea. And it gets narrower and narrower and narrower. And it's a mixture of inflammation here of the epiglottis and then the formation of the pseudomembrane. And in many cases, that would end up closing off the epiglottis, closing the top of the trachea, and would actually form a sort of a valve. When the child breathed, it would close the valve, and the child would not be able to get air and would suffocate. It was a dreadful, dreadful death. And there was very little to be done. There were some doctors who were renowned and very skilled, if you could find somebody, that would go in and would actually kind of remove the top of this pseudomembrane and allow the child some air. In the old days, there was very little that could be done. There were no antibiotics. Corynebacterium does respond to antibiotics. That is one course of action. But the much more important is that this very good vaccine was developed. And the vaccine is not against the bacterium itself. The vaccine is against the toxoid, the diphtheria toxoid. That is the exotoxin, this toxin, this virulence factor that is produced as a result of its acquisition from viruses. And as long as that the diphtheria lacks that virulence factor, the body can deal with it quite easily. It's easily dealt with by the immune system. But if it has this virulence factor, then it proceeds to cause this particular disease syndrome. Any case, nowadays, this diphtheria toxin, this diphtheria vaccine is given as a matter of course. and it's given together with a tetanus toxin, and that is the DTaP vaccine. So nowadays, diphtheria is really relatively uncommon. Okay, so speaking of diseases, upper respiratory tract diseases in children, actually, as any of you have children will know, one of the commonest bacterial diseases in children arises very often as a result of cold or a flu in a child. And it is the production of infections of the sinus and of the ears. Now, sinusitis occurs, all of the things I'm talking about can occur in adults as well, but are particularly common in children. And in children, ear infections, they are predisposed to ear infections. And one of the reasons for that is the structure, the anatomy of the ear itself, and especially of the middle ear. So our ear is divided into three portions. There is an external ear canal, which is closed off by the tympanum on one side. The tympanum is the boundary to the middle ear here, and the middle ear contains these three little bones, which conduct sound from the tympanum to the inner ear here. And the middle ear, because it's a gas-filled chamber, which is almost entirely enclosed, it has to be adapted to adjust to changes, especially in atmospheric pressure. And it does so through this canal here, which is the Eustachian tube. And there are mucose clearance mechanisms as well, but this is effectively an open tube through which air can pass to equalize pressure. In children, this Eustachian tube is extremely narrow, and any inflammation, either down below in the throat during the course of a cold, maybe with a bacterial infection of the throat, this Eustachian tube can be closed off very easily. It's also a passage to colonization by bacteria, and any inflammation that results closes off that Eustachian tube very quickly. And what you now have is, if that is the case, you have this entirely enclosed space here, which can be very rapidly colonized by bacteria, becomes inflamed, produces its own mucus to try and flush this out. But of course, it's got nowhere to go because the Eustachian tubes are closed. And this very rapidly produces an increase in pressure inside the middle ear, which presses both on the tympanum and then on the inner ear as well. In very, very, very severe cases, this can progress to an invasion of the inner ear by bacteria, but this is not common. But it can happen. This middle ear infection is called otitis media. You can also have infections of the external ear canal, and that's otitis externa, which are usually less serious because this is a wide open canal. And although children can get severe earache from otitis externa, you don't have the problems of damage caused by this pressure. So here, for example, this is looking at a child with otitis media. We're looking through the external ear canal at the tympanum here. You can see the little bones here of these stapes here, which connect to the tympanum. And you can see this bulging tympanum. Intensely painful. This is a severe earache. The terrible, terrible feeling of pressure causes huge, huge, huge distress to the child, often accompanied by a high fever as well. Treated with anti-inflammatories and, of course, with antibiotics as well. This usually produces a quite rapid resolution. But unfortunately, because of the child just developmentally has a narrow Eustachian tube, often this recurs. It's difficult to get rid of all of the bacteria, and it may recur periodically. If it does, often the doctors will recommend that the tympanum be punctured. They do this by putting in a little plastic tube called a grommet, which is put into the tympanum so that the middle ear can actually drain to the outside. It can drain to the external ear canal. And this means you don't have the problems of pressure and everything else, and also provides an efficient drainage. And this, often accompanied by antibiotics and everything, is sufficient to clear the infection from the middle ear. Those grommets don't have to be removed. The tympanum is actually quite efficient at repairing itself, and the grommets just fall out and into the outer canal, ear canal. Okay, so those are typical of otitis, sinusitis, and the otitis. These are typical upper respiratory tract infections progressing through the tissues of the upper respiratory tract, etc. But here is an infection which is potentially extremely serious and has a very, very high mortality rate. And these are, I'll first of all say, I'll describe the disease itself, which is acute bacterial meningitis. Acute bacterial meningitis can arise from several different bacteria, especially bacteria of the upper respiratory tract. But the disease results from colonization of the meninges, the coverings of the brain, and then bacterial spread through all of the coverings of the brain and of the spinal cord. It causes inflammation and then can progress to inflammation of the brain tissue itself, which is encephalitis. These bacterial meningitis has a high mortality rate, so it's the occurrence is a medical emergency, and they are somewhat difficult to treat. It is difficult to get antibiotic into the cerebrospinal fluid, for example, and get it to circulate throughout the meninges. So this is a very, very serious disease. The principal cause of this acute bacterial meningitis is an organism called Niaceria Meningitis. Now, Niaceria, you've probably heard about before. The other Niaceria of interest is Niaceria gonorrhea, which causes the disease of gonorrhea. And like Niaceria meningitis is acquired through the upper respiratory tract. In some people, it is actually a normal part of their microflora and never causes a problem. In others, it appears that it is acquired from outside, that there is actually an infection by Niaceria meningitis. The exact reason why it shifts from colonizing just the surface tissues of the nasal mucosa especially, and then becomes pathogenic is not clear. But what is clear is that there is a distinct shift in the mechanisms of the bacterium. Now, I'll tell you first, this is a rather unusual bacterium because you can see here it is a Diplococcus, but it is a gram-negative Diplococcus. And gram-negative cocci are extremely unusual. The vast majority of cocci are gram-positive. This is not, it's gram-negative. It begins by colonizing the nasal mucosa in the normal way. It just attaches to them. But there is a shift where it crosses over. It begins to make its way through the mucosal tissues and through the tissues of the blood vessels. And it will actually move into the blood vessels, and it will then circulate, and it has a preference for tissues associated with the brain and with the spinal cord. And this is how it ends up circulating in the cerebrospinal fluid and colonizing the meninges. It would stress, it appears that in some people, they maintain populations of Niaceria Meningitis as a normal part of their microflora. But in cases where this blooms, and especially where people become symptomatic for meningitis, those people can become extremely infectious. And there are minor epidemics of bacterial meningitis which move through, for example, army barracks and university dorms and things like this. There's also in Africa, there's a huge belt that's not clearly understood. There's a huge belt all the way across the equator and the subtropics, where bacterial meningitis is incredibly common compared to the rest of the world. And that's called the meningitis belt in Africa. This can be treated with antibiotics, but the best prevention is actually a vaccine. There is a fairly good vaccine which offers protection against this. Now, this acute bacterial meningitis, there are a couple of other bacteria which will produce similar symptoms. And one of them is Streptococcus Pneumoniae, which we will hear about again in a while. So the other is another organism, which is a rather strange organism. Again, sometimes it appears to be disease-causing. It can cause a very serious sore throat. But in other people, it is recovered as part of their normal microflora. And this is Hemophilus Influenzae. It has nothing to do with influenza, except that it sometimes follows an influenza illness as a secondary bacterial infection, but it does not cause influenza. So those two organisms can also, by the similar mechanism, end up causing meningitis. This is how it goes. They have the exact ways, exact reason why they commence this particular route of infection is not known, because this is extremely complex. What they're doing is they're inducing the cells of the mucous membrane to actually take them in and allow passage across the epithelium, the mucous epithelium. And they then end up moving across the endothelium, which is the lining of the blood vessels, and end up migrating specifically to the cerebrospinal fluid. So this is a very complex set of steps of pathogenesis. And the exact reason why they shift from this relatively benign form to this highly virulent form is not understood. The other two, the Streptococcus and Hemophilus, they do the same, adopt the same kind of mechanisms to do so. But Niaceria is by far the more common cause of it. Okay, so here's another of the potentially dangerous childhood bacterial diseases. Now we're moving from the upper respiratory tract down to the lower respiratory tract. The lower respiratory tract, infections of the lower respiratory tract are potentially far more serious. And as we'll see, and this is one of the reasons why there is this very, very efficient clearance mechanism to continually scrub the lower respiratory tract. Here is another of the dangerous bacterial diseases of childhood, and this is pertussis, or whooping cough. And whooping cough is a very, very contagious disease, and it's caused by a bacterium called Bordetella pertussis. It's an aerobic gram-negative rod. Here you can see it sitting here, and it preferentially colonizes the ciliated epithelium. So that it can be all the way down to the bronchi. It can infect the lower respiratory tract. And it also produces a toxin. It produces an exotoxin. And this has a specific activity. It paralyzes the ciliated cells, and it allows the bacterium to form dense colonies throughout the ciliated epithelium. And at the same time, it causes inflammation of the bronchi, and it produces this vicious cough. What happens is that the child will cough repeatedly, and in what is referred to as a paroxysmal cough. They cough and cough and cough and cough and cough and cough without taking a breath, over and over and over again, until they've completely exhausted the lungs. And they then attempt to breathe. And what's happening is they're trying to breathe through inflamed, narrowed bronchi and trachea. And so they go, when they breathe in, they give this terrifying whooping sound. And they often have this panicked, choking look on their face because they're getting so little air. And it is a really terrifying thing to watch. And the child can actually die during a paroxysm. And they could die as a result of just the complete lack of oxygen, of anoxia, because of this. Nowadays, it is very easily treated with antibiotics. In the old days, it was another of the great childhood killers. But nowadays, first of all, it can be treated with antibiotics. But second of all, there is a good vaccine as well. And this is one of the standard childhood vaccines. It is against pertussis. So this is also usually given. The vaccine, again, is against the toxin. If the bacteria lack the ability to have the toxin be effective, they are unable to produce their complete pathogenesis, and they are easily dealt with by the immune system. So this is part of this Tdap vaccine, which is routinely given. And this is tetanus, diphtheria, and pertussis. They are given together. And it's a very good vaccine, very, very effective. So you can see, okay, this is kind of an object lesson, right? You can see the rates before, this is from 1990. This is the incidence of whooping cough per 100,000 of population in the United States. And this is actually, it says here 1900 to 2010. In actual fact, this says from about 1990. I think that should say 1990, not 1900. But in any case, you can see the rates are relatively low. This is because of the prevalence of use of antibiotics. And then there is something in 2000s here, which I suspect is an outbreak, which is brought under control by widespread vaccination. And then we have a period when vaccine reluctance begins. And you can see now that the incidence begins to increase again. Another example of the impact of vaccine reluctance. This is a very dangerous disease. This is not something you want your children to be exposed to. And this vaccine is essential. It gives protection both against diphtheria, which is potentially fatal, and against pertussis, which, even if it wasn't fatal, is an extremely unpleasant disease for your child to have to live through. There is some indication that this should be given a 2-shot vaccine. Okay, so let's just have a look now at one of the greatest killers of humankind, which has been around, been in the human population probably since we we became human. I say that because this disease of tuberculosis, one or other form of it, is present in many mammals and is certainly present in other primates, although there are different species of the bacterium. We have almost certainly had tuberculosis in the population since we evolved, and certainly even in fossils, some fossils of fossil humans have been found with evidence that they actually had tuberculosis and may have died of it. We do know, if we go back several thousand years to the ancient Egyptians, for example, that tuberculosis was present, that we can see it in some of the mummies. There is tuberculosis, evidence of tuberculosis there. So this is a very ancient disease, and it has actually been a major, had a major impact on human population, on the size of human populations. Currently, believe it or not, of our population, current population of humans is about 7 billion. About 2 billion people, is estimated, are infected with tuberculosis. They are not all necessarily ill, or not ill currently, and but 2 billion people are infected with tuberculosis and have at least the potential for suffering severe illness and certainly for becoming infectious. So this is caused by, the disease is caused by the organism Mycobacterium tuberculosis. Now, Mycobacterium tuberculosis is closely allied to the gram-positive bacteria, but it is not a gram-positive. It is one of these strange bacteria which produces a very thick coating in its cell wall of mycolic acid. Mycolic acid is a wax and repels aqueous stain. So they're very difficult to stain. You can't put them through a gram stain because they don't respond properly. Instead, you have to use a special staining technique, which you may have heard about in lab, called the acid-fast stain. Mycobacteria, there's a lot of many species, and not all of them are pathogenic. We have some which are commensal as well. But there are two which are of particular importance to human beings, and one is Mycobacterium tuberculosis. The other one is Mycobacterium leprae. And the Mycobacterium leprae, although it manifests as what looks like a skin disease, is actually largely an infection of nerves and of nerve endings. But that's not our concern at the moment. We want to put our attention to Mycobacterium tuberculosis. Here is a smear. This is probably a sputum that's been obtained, full of white blood cells, but also full of these red cells here, which again, they tend to palisade. They lie next to one another like this. And you can see them here. You have to look quite carefully to see the individual cells, but these red collections here, these are collections of acid-fast Mycobacterium. And this is diagnostic in a sputum. This would be diagnostic of the disease of tuberculosis caused by Mycobacterium tuberculosis. In general, it is acquired through aerosolized droplets, but it is actually not particularly infectious. It tends to be picked up from prolonged and intimate contact with somebody that has the disease. It's not always. Obviously people can inadvertently pick it up outside, but it tends to move in family groups and things like that. So people that are constantly in contact with another, they do tend to spread it to one another. It has a very strange life cycle. It has been in the human population for so long that it has formed a very intimate relationship to the cells of the immune system. And in fact, there are whole arms of our immune system which appear to be have evolved specifically to recognize the presence of Mycobacterium tuberculosis. They don't always do it perfectly. So what happens initially is if you breathe in Mycobacterium, then and it is able to set up an infection. First of all, it infects the tissues of the respiratory tract and is immediately recognized by the first arm of the immune system. And the major way in which it is recognized is macrophages, which are white blood cells, recognize the presence of the bacterium and actually eat it. They phagocytose it. Now, the normal course of events for other bacteria, macrophages do this the whole time. They see the other bacteria and they go and they eat it. That's the function of molt and everything else. The macrophages march out, they swallow bacteria, they normally digest them, and then they actually show parts of the bacterium on their cell membrane. They're kind of advertising the presence of an invader by showing bits of it on their cell membrane. That's the normal function. But for Mycobacterium tuberculosis has evolved so that it suppresses that mechanism. And it actually survives inside the macrophage and begins to breed. Now, there are still ways in which the immune system can recognize that a macrophage has been infected by Mycobacterium, but these are rather imperfect. And a very complex dance starts between the cells of the immune system. So let me just, yeah, okay, we can just, here's the cells being inhaled. They are actually ingested here. They are ingested by the macrophages, and but now the macrophages actually fail to kill off the Mycobacterium inside them, and we now have the macrophages going around containing these potentially dangerous bacteria. So the response inside the lung tissue especially is that they now start interacting with other cells of the immune system. And the other cells begin to surround these infected macrophages. Here they are here. These are macrophages and other cells of the immune system, which have recognized the presence of infected macrophages. And they come to surround them like this. And they form a spherical cluster around these infected cells, which is called a tubercle. This is the reason that this disease is called tuberculosis. Because when you look at the lungs of somebody who's been severely infected, you'll see that the lungs, all through the lungs, there occur these clusters of cells surrounding the infected cells, these tubercles. Now, as this progresses, if this now carries on just like this, eventually the body can form a fibrous sheath all the way around. The problem is these are extremely long-lived, extremely long-lived. So people can maintain them, in fact, for their whole lives. In many cases, in the majority of cases, in fact, with a mild infection of tuberculosis, with only a few of these tubercles, they are simply maintained as a latent infection. People never know that they actually have the disease. But it is possible for this protective mechanism to break down. When it has a sheath around it like this, it's called a granuloma. And they appear very clearly on X-ray. So this is how we know that many, many people have granulomas and have never had symptoms of tuberculosis. But it is possible for these granulomas to break down. Exactly why is not known. It appears that these infected macrophages, the bacteria suddenly become extremely active and seem to become extremely virulent. And this granuloma breaks down. And the bacteria, the macrophages are killed in the center, and the bacteria are released en masse into the lungs and into the blood system of the lungs. From the blood system of the lungs, it may in fact spread through the rest of the body, but it is primarily an infection of the lower respiratory tract. And it can, once these granulomas break down, it can spread extremely rapidly through the rest of the lungs and ultimately become fatal. The maintenance, the one thing we do know is that the maintenance of this granuloma appears to be an immune function. It is a result of an interaction of this whole structure with the immune system. And the destruction of the granuloma, the loss of the granuloma, and the release of the bacteria is due to some interaction failing and allowing the granuloma to disintegrate. So here is you can see here in this colored X-ray, this here, all of this tissue here is infected with tuberculosis. It is an extremely toxic, very, very debilitating bacterium. Quite apart from the fact that it begins to impair respiratory function, it also produces exotoxins, which have systemic effects throughout the body. And it is an extremely debilitating disease. People become extremely fatigued, extremely emaciated, and in people with disseminated tuberculosis, this can proceed quite rapidly and quite rapidly result in death. So there are various ways in which tuberculosis is diagnosed. First of all, symptomatically. It has some classical symptoms like night sweats, fatigue, loss of weight, unexplained loss of weight, etc., etc. Second, of course, is X-ray. It appears quite clearly on X-ray, but it can also be tested for with a serological test. The classic one is MAN2 test, and what they do is they inject under your skin a small amount of tuberculin, which is derived from dead tuberculosis bacteria. And if there is a positive skin reaction like that, it's an indication that you have been exposed, at least exposed to tuberculosis and have produced antibody to it. So right up until the 1930s, there was very little that could be done for people who were suffering from symptomatic tuberculosis. The classic treatment was to put people into circumstances where it could be hoped that an active infection could be pushed back into this situation. Where you could actually get tuberculosis back into a granuloma and push infections into a latent state again. This was done by, first of all, the major thing they should say was bed rest, to try and limit activity, limit respiratory activity especially, which tends to spread the bacterium. But also they would sometimes come in, for example, and collapse a lung. If one lung like this was severely infected, they would collapse a lung, or they could collapse the lobe of a lung and allow the lung lobe to consolidate. It would simply collapse, and then the tissues would just all stick together, and it would eventually just form scar tissue and enclose the tuberculosis, the mycobacterium in that. But there was not much that could be done right up until the 1930s, when the first antibiotics specifically designed for mycobacterium came into use. These were initially literally miracle drugs. Tuberculosis was incredibly common. It was common in all age classes, all social classes, but it was especially common under crowded conditions of poverty and everything. And there, for example, one of the treatments was to send people to a TB sanatorium. This accomplished two things. It first of all put people into healthy conditions, a healthy environment, where it was supposed with good nutrition and everything else, their own body's mechanisms would hopefully overcome the infection. The second thing was it enabled people to rest. But the third most important thing was it isolated people who were potentially infectious from the general population. And this idea of quarantine and of isolation became very prevalent. All of those mechanisms had some success, but not much success. With the advent of antibiotics, many of those people were successfully treated, and within a short time of treatment, their symptoms would resolve to the extent that they were no longer infectious and they could return to the general population. And it became standard, in actual fact, that tuberculosis, if rapidly diagnosed, could be treated on an outpatient basis. So very soon after antibiotic treatment started, people would be non-infectious and could return to the general population. The incidence of tuberculosis plummeted worldwide as a result of these antibiotics. But unfortunately, this bacterium is able to quite rapidly develop resistance to antibiotics. And most of the early antibiotics with early sulfur drugs and things like that are completely useless against modern strains of Mycobacterium. And there has been a continual battle, pharmacological battle, to develop new antibiotics to which Mycobacterium is not resistant. And it has become an increasingly desperate battle, because we now see that there are Mycobacterium strains which have arisen, which are resistant to virtually all the available antibiotics. And in many parts of the world, the tuberculosis epidemic, ongoing epidemic, is becoming more and more serious. There is nowhere where this is more true than in Africa. And the reason for that is that throughout Africa, the AIDS epidemic is still raging. And many people are under treatment with early AIDS anti-HIV medications, which are very limited in their effectiveness. And many people are not being treated at all. And the HIV disease is a long-term disease. It takes, people are ill for many years, and for many years have an increasingly impaired immune system. Well, let's go back here and realize that TB is also extremely common in Africa, but under normal circumstances, up to 90% of people infected with tuberculosis never proceed to symptomatic disease. Why? Because their immune system is maintaining a latent infection in this form. They have granulomas, which have potential to produce disease, but in most cases, the immune system maintains the granuloma and keeps the disease latent. Now, simultaneously, we have the HIV epidemic, where people's immune systems are beginning to collapse. And one of the early symptoms of that collapse is that these tubercles and granulomas are no longer maintained by the immune system, and they break out. And people who simultaneously have AIDS or HIV infection and tuberculosis are the source of massive cross-infection to the general population. Not only that, but these people, they maintain a tuberculosis infection together with the HIV over a very long period of time. And if they are in fact treated with antibiotics, they act as almost as petri dishes for the development of resistant strains. So a major contributor in Africa, at least, a major contributor to a current huge wave of tuberculosis is the AIDS, the parallel epidemic of HIV infection. Now, unfortunately, the ideal thing would be, of course, to have a good vaccine against tuberculosis. There is a vaccine against tuberculosis. It's called BCG, and that stands for Bacillus calmette-guerin. And Bacillus calmette-guerin is an actual bacillus, but it's an artificial bacillus. It's an attenuated bacillus, which was developed from a strain of tuberculosis in cattle called Mycobacterium bovis. We can actually acquire Mycobacterium bovis as an illness, but along with Mycobacterium tuberculosis, but it is much less common. But the bacterium can be modified to form a vaccine, and it's called the BCG. It is usually administered to children. In Africa, it is commonly administered to children, and I think in Europe as well, and in many parts of the world. But it has disadvantage. It works quite well in children, but it loses its effectiveness around puberty. And it doesn't appear to be renewed. You can't boost it for some reason. And so it has limited, but it's useful for limiting infection in children. That is true. There has a disadvantage. Once you've got it, once you've had that vaccine, you can't have one of these serological tests, right? Like the MAN2 test. You can't have that because you will simply react very, very strongly to it. So that is another disadvantage. So the BCG vaccine, for example, is not given in the United States, where childhood tuberculosis is very uncommon anyway. And but in much of the rest of the world, it is given, but this is of extremely limited use. And a really good, effective anti-Mycobacterium vaccine is still of the subject of research. Okay, so let's pass on now to another of the really serious lower respiratory tract infections. And this is another Streptococcus. This is Streptococcus Pneumoniae. Now, you've heard about Streptococcus Pneumoniae before, but in a completely different context. And that was when we talked about bacterial genetics. And I told you that this was the subject of research by Griffith. And what he was interested in was the fact that there are two strains of Streptococcus Pneumoniae. And there's one which is pathogenic and one which is not pathogenic. And the virulence factor in the pathogenic one is that it produces a capsule. So this is a gram-positive coccus. It can form short chains, but it very often is in the form of a diplococcus, 1, 2, 1, 2, 1, 2. But it can form short chains as well. This is typical. This is a capsule stain for a Streptococcus pneumoniae. And you can, first of all, let me show you here. This is a gram stain, and the typical gram stains, the gram positive. Here they are here, quite easy to see. But if we do a capsule stain, you can see the cells, the darkly stained cells in the middle, this light pink background, and then a clear area surrounding the cells. And that is the capsule. And the capsule is really important because it's a virulence factor. It allows this bacterium to adhere to the cells of the lower respiratory tract. and it protects the bacterium from immune activity, especially of the white blood cells. They just are unable to see the bacterium because it's hidden underneath a thick, slimy capsule. This is why this is pathogenic, and a non-encapsulated one is not pathogenic. So, Streptococcus pneumoniae, it is the classic cause of pneumonia. Pneumonia is a widespread bacterial, or it can be viral as well, infection of the tissues of the lung itself, of the alveoli, which causes inflammation in the alveoli, and it causes the mucous membranes to produce abundant mucus, which pools and consolidates in the alveoli, forming a consolidation of thick mucus and in which bacteria rapidly breed. So it's this kind of purulent mucus inside the alveoli themselves. And of course, what that is going to do is it's going to fill the alveoli, and they're not going to be able to accomplish oxygen exchange, oxygen and carbon dioxide exchange. So the infection can be widely disseminated through the lungs, or it can be localized. And it appears on X-ray as these areas of consolidation. And this is an entire lobe of the lung here, which has been infected and consolidated. There is a little bit of other infection in this patient, but this patient is extremely ill. Along with this, obviously, this is severely impairing when it's this serious, widespread. But the infection itself produces other symptoms, systemic symptoms of illness as well. So it is extremely debilitating. Now, they used to call this, it's probably not politically correct anymore, but they used to call this the old person's friend. The reason for that was this pneumonia is typical pneumonia of somebody who's very elderly, probably with an impaired immune system, who has been lying down and not exercising and not clearing the lungs properly. Now, that is typical of a very elderly person who is debilitated and invalid. And very, very often, the end of that story was the development of a streptococcal pneumonia like this, which it's referred to as a pneumococcal pneumonia, because this is caused by Streptococcus Pneumoniae. So do you call it a pneumococcal pneumonia? And that was the classic thing. This was what carried this suffering old person off. And the trouble with that kind of attitude is, in fact, we ignore the fact that this pneumococcal pneumonia can occur at any age, does not necessarily occur in people who are already debilitated. It can result, for example, from severe influenza or something like this as a secondary infection. And in fact, it's still responsible for 20%. 20%, that's a lot of childhood mortalities. Fortunately, there is, in fact, a very good vaccine. And this is one vaccine which you should keep careful note of. It should be given, it can be given in childhood, but it can also be given later in life. And it is one of the vaccines which you should boost as you get older. Make sure that, that you have. There are also a couple of different vaccines, I should add, because there are a couple of different strains that have been identified. But this is definitely something that you should discuss with your medical practitioner and make sure that you maintain your pneumococcal vaccinations. So that's the most important. That's Streptococcus pneumonia. That's the most important and one of the most virulent of the pneumonia causing bacteria. But there are a number of others, and some of them are very, very definitely associated with healthcare settings. Streptococcus pneumoniae, actually, you can recover it from most people's microbiota. So many people regard Streptococcus pneumoniae as being an opportunistic infection. It's a part of the normal microflora and takes advantage of situations where it can become pathogenic. These other ones are also probably most of them opportunistic in that they can be found in other normal settings. But they are also, it has been shown that they can be easily transmitted in healthcare settings. So these are really transmissible. Pneumococcal pneumonia tends to be regarded as kind of an intrinsic thing arising from our own microflora. The first of these healthcare-acquired pneumonias to think about is ordinary old Staphylococcus aureus. And Staphylococcus aureus is part of our skin microflora. But under certain circumstances, especially in circumstances where people are immune compromised, the very elderly, people suffering from HIV disease, etc., Staphylococcus can, in fact, cause a pneumonia, and it's a very dangerous pneumonia. It's called a necrotizing pneumonia, which means that it results actually not just in that consolidation of the lung tissue, it actually ends up degrading the lung tissue, causing lung tissue to die, to necrotize. So it's a very dangerous. Fortunately, it is not particularly common, but it is certainly very well known. The next one is Klebsiella Pneumoniae. Klebsiella is another organism which can be recovered from people's microflora, but it again can be an opportunistic infection, especially in immune compromised, and is particularly in healthcare settings amongst the very young. It forms, unfortunately, periodically, it forms epidemic infection in nurseries, for example, in hospitals where there is some breakdown in normal hygiene procedures, especially mask wearing. It appears that it can be spread both by aerosol from healthcare workers, but also by hand and dealing with the children. And it's in children, in infants, it's an extremely serious pneumonia, Klebsiella pneumoniae. It does also occur in people with impaired pulmonary function and in the elderly, but its most serious impact is on infants. The last one, this is Pseudomonas Aeruginosa, and this is really is an opportunistic infection because Pseudomonas Aeruginosa is actually an environmental bacterium, very easily isolated from soil and this sort of thing. So it occurs widely in the environment, but in severely immunocompromised patients, and especially people with HIV disease and in burn patients, Pseudomonas aeruginosa is a very dangerous opportunistic infection. In burn patients, it often will infect the burns themselves and produces a characteristic kind of green sheen, which fills healthcare workers with dread because people seldom recover if they develop that infection. But it can also produce a very serious pneumonia. So I think this is the last one which I'm going to tell you about today. And I've actually mentioned this one before. This is Legionella Pneumophila. And Legionella Pneumophila is really interesting because it's a relatively recently discovered disease. It was only recognized in the 1970s. And its etiology elucidated in the 1970s. Legionella Pneumophila is a gram-negative rod. Again, it's really an opportunistic pathogen because it occurs normally as part of aquatic flora. And it can easily be isolated from streams, from soil, other places, but of course in relatively low numbers. But it tends to occupy water which is standing, ponds, pools, this sort of thing. And it grows as part of the aquatic community, especially it's associated with certain amoebae and things. And under certain conditions, it can reach very high population numbers. And unfortunately, one of the places where it grows are air conditioners and units like that, where they have drip trays, where they have trays which collect condensate from the cooling systems and everything else. In the old days, those drip trays were often open, relatively open. And when the air conditioner kicked into action to pump air through a building or whatever, they would stir up the water and catch droplets. And the droplets would be spread through the air conditioning unit, and along with them, Legionella Pneumophila. The first time this was recorded as happening was in Philadelphia at a convention of the American Legion, these elderly gentlemen who gathered in a hotel, and they spent days talking to one another in this hotel, and it was hot, and they had the air conditioner on, and it was blowing Legionella Pneumophila all over them. And there was an epidemic which ran through the participants a couple of days later, and many of them died. And it was a very spectacular event. That's where it gets its name, Legionnaire's disease. That's where the name of the bacterium comes from the Legionnaires. It's Legionella. And it produces a very serious pneumonia. In young people, it can also produce the pneumonia, but in people with a very healthy immune system, it produces a lesser disease called Pontiac fever, which is more like influenza. But true Legionella pneumonia is extremely serious and has quite a high fatality rate. And it remains an important bacterium. We had an outbreak recently, 2 years ago or so, in the Bronx. Now, modern air conditioners take this into account. And modern air conditioners isolate the air conditioning mechanism from any drip trays or anything like that, so that this can't happen. But there are many old air conditioning units which are still in use, and there was one in an apartment block in the Bronx. And interestingly enough, it was not a matter of it blowing infectious air through the building. It was blowing infectious air to the outside of the building. And everybody who acquired Legionnaire's disease from that had been a passerby on underneath the building. And a couple of people actually did die as a result. So it remains an important disease. Okay, so that's it for our airborne bacterial diseases. And we'll move on to another group next week.